Now next is the incubation period of the titration. Incubation period method for the titration of the different kind of the viruses. What is the incubation period? Actually, the incubation period is specific kind of the period which is the period between the inoculation and the first sign which is shown by that specific kind of the animal. It is called the incubation period. It is based on the fact that some kind of the viruses have the different kind of the time for the incubation, different time for the seeing the first sign of that viruses. Of course, there should be some precise kind of the relation between the amount of the inoculum and the incubation period which is used. If the amount of the inoculum will be the more, then the first sign will be appeared very quickly. If the amount of the inoculum will be smaller, of course the sign will be not seen very quickly. So for the incubation period method of the viral titration, we see it in detail that how we can just examine and observe our results. First of all, it is the most successful method for the viral titrations. It was first seen by the rabbit papilloma viruses which were done by the Brian and the Beard, and they were investigating about the rabbit papilloma virus, and they have seen that the incubation period method can be used for the viral titration, and this is the very important method. The most important observation was the time which was used between the inoculum and the appearance of the papilloma. The rabbit papilloma virus is shown its clinical signs on the skin, and they were very surprised to see that different time was seen by the, and time was dependent on the amount of the inoculum. The time between the inoculum and the appearance of the papilloma on the skin was related to the amount of the inoculum. So here they have seen the relationship was expressed in an equation which is shown here. T2 minus T1, which is equal to the B into log C1 and the log C2. A plot of the T versus log C will give us the straight line as it is clear in the here. You can see that incubation period, how to describe the incubation period here. First of all, the time 2 is the T2 is the time 2 and T1 is the time 1. First of all is the inoculum and how much the concentration of the inoculum was given. Then the time 1, first the clinical signs which were appeared after the inoculum was injected inside. And the plot here, the incubation period in the days, which is about 10, 15, 20, 25. And then here is the amount of the inoculum. So the plot of T versus log C will be as the straight line graph as is clear from the here. It should be appeared, you can see it, and it is the straight line graph should be considered. However, this the inoculation period, which this can also be in the specific kind of the graph which is shown in the next slide. Here you can see the units of the antigen which are present here. You can see the units of the antigen and then the number of virus lesions which are present on the vertically, and then the units of the antigen which are on the horizontal. So here you can see it just simply, if we can take it 0, 4, 8, 18, 16 and 20, 24, and then the number of lesions which are shown by each virus will be in the form of the hundreds, 100, 200 and 300, and you can see that how much lesions. And in this way, if the at some specific type of the virus, first it is going to increase, but at one special time it will be, they will be in the straight line here. For example, in this case we can just take it at 400, and there will be no much effect of the increasing of the units infectivity or the antigen. Here you can see it just in the laboratory if we are working on some specific kind. So here is the virus dilutions which are shown here, 1 into 2, 1 into 8, 32. It will be the positive, positive, positive. But at this dilution, which is very smaller dilution, 1 into 1 to 8, still the results are the positive, and you can see in the plates too. However, at the 1 into 5, 1, 2, it is will be used, the dilution is too little, and therefore it is very small here. And then this is the positive, the dilutions. There are many viruses, and then you can see the negatives. This is an example of the red blood cells in the viruses. You can see in this way that incubation period method how it acts.